All right. Hey everyone, welcome to topic 9 review session. Gabe and I are going to go over DNA replication. This is only a 2-person review session. So we're just going to go over some slides, and then we'll have we'll have time if you guys have problems, questions or whatever you want later. And then also Gabe, I don't know how we wanted to.
You can do halfway.
Sure. All right, so we're going to go over DNA RNA structure. So the DNA and RNA, they're types of nucleic acids, right? And the monomers for nucleic acids are nucleotides. And the structure of nucleotides are, well, they're composed of 3 things. They're composed of a phosphate group, a sugar group, and a nitrogenous base. Looking at this picture, is this, well, I mean, there's only you here, and then I don't know if I can hear Tori, but is this a DNA or RNA nucleotide?
I think it'll be DNA.
Yes, DNA. And Victoria also said it in the chat, DNA. So it's DNA, right? Because why is it DNA? So it's DNA because there is not an OH group on that 2' carbon. So this is the sugar group, right? The way that we label the sugar group is that this is 1', 2', 3', 4', 5' carbon. And in RNA, there is a hydroxyl group on the 2' and the 3' carbon, but in DNA, there's only a hydroxyl group on the 3', right? So here there's only a hydrogen. Okay. And then this nucleotide, this is a purine, right? Because purines are ones with the nitrogenous bases that have 2 of these hexagon structures, and then pyrimidines have 1. And then the next nucleotide is added here at that 3' carbon where the OH is. Okay, so these are the different DNA nucleotides. You have adenine, guanosine, thymine, and cytosine. Adenine and guanosine are purines, and thymine and cytosine are pyrimidines. And this is one way that you can remember it. Pure as gold purines include adenine and guanine. RNA nucleotides, they include all of them except for thymine, and RNA has uracil instead of thymine. This is just basic DNA structure stuff. Double-stranded DNA, they are anti-parallel, which means that, so this is the 5' end at the top, 3' end at the bottom, and they base pair with another DNA strand, and it's anti-parallel. So the 5' end is here, so the 5' end of the other one is on the opposite side, right? So they're flipped, and then they hydrogen bond. And the different nucleotides in the DNA are bonded through phosphodiester bonds, right? So this is a nucleotide. This is the phosphodiester bond. This is the next nucleotide. This is the phosphodiester bond. So pretty basic. The way that nucleotide bonding occurs is that this OH, right, on that 3' carbon of that nucleotide that you're building off of, this basically attacks one of the first phosphate of a next nucleotide, which then causes the phosphodiester bond. And you don't need to really well, yeah, you just need to know that this OH binds to that first phosphate group, and then these 2 phosphates are released, and then the breakage of this phosphate bond to these other 2 phosphates is what causes the new bond to form. But yeah.
it's good to have this visual so you know that you're always adding nucleotides to the 3' end.
Yeah. Um. We already sort of went over this, but this is the sugar group in DNA. This is deoxyribose. And then this is showing the different carbons. The 1' is the carbon where there's a base on this side. So if you want to attach the base, this is the 2' carbon where in an RNA you'd have another OH group. This is the 3' carbon with that OH group that is used to add another nucleotide. And then this is 4', and then 5' is where the phosphate is attached. Yeah. And there are different numbers of hydrogen bonds that occur between the different nucleotides. So when T pairs with A or U pairs with A in RNA, you get there are 2 hydrogen bonds that form between the bases. And then when G pairs with C, you have 3. And this is significant because in DNA structures that have more GC pairs, there's a stronger binding, right, in the DNA because there are more hydrogen bonds. So knowing this, I won't, since you're the only person here, basically, I won't ask the question, but is this the same one?
I think it is.
It is. Whoops.
Yeah.
All right. Whoops. Okay. Well, this is supposed to be different. I'll change that later. But basically, the idea is that if there were, let's say that instead of like these 3 ATs, there would be GC pairing. This strand would need more heat to separate because there are a lot more hydrogen bonds holding it together.
Oh, wait, so like the more like AT like bonds are, like easier it would be to break apart.
Exactly. Exactly, because of the number of hydrogen bonds occurring between the bases. Okay, so, all right, for these next questions, we'll go over whether this occurs for DNA, RNA, or both. So made up of nucleotides, both. Contains ribose as its sugar.
Oh, wait, only DNA.
RNA, remember DNA deoxyribose. So yeah. And then has a sugar phosphate backbone. Both. Yeah, right. Both. The only thing that differs between them is just the sugar is a little different, and then the base in the RNA, it uses uracil instead of thymine. Oops, and that's the next one. So RNA uses uracil. RNA single-stranded. Contains an OH bond with a 2' carbon.
DNA.
At the 2' carbon. Remember which one has the 2' carbon? This is 2'.
Oh, so RNA.
They both. So yeah, both DNA and RNA have that 3' OH, but only RNA has the 2' OH. Oops. And then contains phosphodiester bonds. Both.
Yeah.
Yes. Uses thymine. Contains deoxyribose as a sugar.
Yeah.
Double-stranded. Only found in the nucleus. Can pair with itself.
Or both. Like,
Compare. Sorry. A single strand can pair with itself.
oh, okay, so that'll be
RNA. Yes.
compare. I used to like compare with itself. I just forget DNA both things.
Yeah. No, makes sense. All right. So, why do you think it's a good thing that our information storage unit, which is our DNA, is made up of DNA nucleotides and not RNA? So this has to do with that extra OH group on the 2' carbon.
Because like it's more like balanced.
Yeah, more stable.
Stable.
Yeah, exactly. So that that extra OH on the RNA nucleotide, on the 2' carbon makes RNA more unstable and
Which is good
reactive.
because RNA is designed to be like, once you use it, you quickly degrade it.
Exactly, right? So, because DNA does not have the extra OH group, it's more stable than the RNA, which is a good thing because you want your DNA to remain there and not to get degraded. All right, so this is just like some math. I'll just like quickly go through it. A particular piece of double-stranded DNA contains 134 adenine bases. How many thymine bases are there? How many guanine bases are there? And how many cytosine bases are there? So I'll just like walk through this question a little. So, what does adenine pair with? Exactly, adenine pairs with thymine. So if we know that there are 134 adenine bases, how many thymine are there? 134, yes.
34.
Yes. Okay. Knowing this, is it possible to know how many guanine and cytosine bases there are?
No.
No. Exactly.
Total amounts of like bases there are.
Exactly. We don't know the total amount of bases, so we can't tell how many guanine and cytosine bases there are. Exactly. All right, so this is basically same question, but instead of saying a number, we say a percent. So we say a double-stranded piece of DNA contains 14% adenines. What's the percent of the other nitrogenous bases?
So this we just subtract that by 100, it would be like 86%.
So if there are 14% adenines, what percent thymines are there?
Thymine, so that would be 14%.
Exactly. So 14% plus 14% is 28, right? So that means
That may be 36% of the C and 36% of the G.
exactly, right? So now that we're talking about percentages, we can make a statement about the whole strand, right, that we're talking about. But if we were given just a number and we don't know how many in total there are, then we can't make any decisions on the other bases, just whatever adenine pairs with. If there were a double-stranded piece of DNA that contained 30% adenines instead of the 14%, would it be easier or harder to separate the DNA? So this is like talking about the
They have only 2 more.
exactly, yeah, right. So the strand that contained 30% adenines, it would be much easier to separate because the AT bonds, right, those contain 2 hydrogen bonds and not 3. So, yep, good. All right, an mRNA contains 40% uracil. What is the percentage of the other nitrogenous bases? Can you tell from that?
No, I don't think you can because it's an mRNA.
Exactly, it's single-stranded, right? So because there's no complementarity and like there's
Base.
exactly, you can't say even with like adenine, you can't say adenine is 40%, right? Because it's just a single strand. It could be 40% uracil, 3% adenine, yeah, whatever, but you get it. All right. Victoria, we are checking the chat, so if you have any questions, feel free to put it in there. Okay, I'm gonna move on. All right, do you want to look something there?
Okay, so DNA replication this is like a general overview of it. So DNA replication is a semi-conservative model, which means that in the parental strand seen over here, half of its DNA is going to end up in the daughter strands of the DNA. And then in the next generation, you can see that it continues on where half of it goes to the one half and used as a template, and half is used as a template for another strand, and the same is in both. So in each, the semi-conservative model basically just means that each one, yeah, so the semi-conservative model just means that half it ends up in one strand, the other half ends up in another, and they use the strands as a template for complementary base pairing. So the origin of replication is places in the DNA where DNA replication is going to start. And all along a particular chromosome, there's going to be a bunch of origin of replications. And each one is going to have this bubble open up where helicase is going to go and open up in both directions. And eventually these bubbles are going to like quote unquote like meet each other until and at that point they'll become two separate strands. Yeah, since there's billions of base pairs, you can't just have one origin replication. It would take way too long. And then for circular DNA like bacteria, there's only one origin, and replication goes in both directions bidirectionally. There's two replication forks. Yeah, so there's two replication forks, one right here and one right here, left and right at each bubble. And we'll go more into detail about like each side, like which one's the leading strand and lagging strand. But yeah, the end result is that the bubbles all meet each other, and then there's two DNA strands. Any questions so far? Okay. So the main point of the beginning of DNA replication is that one conceptual thing you need to know in order to understand like the leading and lagging strands is that DNA polymerase needs a 3' end to add on nucleotides, and it can't start DNA synthesis on its own. So it needs to have an existing 3' end. So a short RNA primer is added by this enzyme called primase. And so as you can see here, this is like the RNA primer added. And these 8 to 10 are error prone. So later they're going to be removed. But first, DNA polymerase is going to add on to this 3' end. And then it can keep growing this way. But as we'll see, and then later on, the RNA will be replaced with DNA. But as we'll see, because of the way the origin of replication opens, one direction, it's not going to be able to continue on like this if it's not opening in that direction, which leads to a leading and lagging strand. And so for a lot of like your problems, you're gonna have to label 5' and 3' end. And so you'll have to know which one. You either have to have like a diagram with like pieces of DNA or like 3' or 5' ends or leading or lagging in order to fill in another part of that. You can't just figure it out from like none of them. And the direction, if there's nothing here, you'll have to just draw your like 3', 5' and label stuff. So the directionality is really important. You need to see whether when you're going in one direction, it's 3' or 5'. These are the parent strains. And if you're going in the 5' to 3' direction of the fork, then you're in a leading strand. Otherwise, it's a lagging strand, because as you add on in this leading strand, for example, if this is 5' and 3', then this would be the 5' end of like the strand that's going to be built, and this would be 3' end, so it can add on continuously. Whereas it's still, for this one up here, 3' end is over here, so it's not going to be, as it opens up, then more and more of the 3' end is exposed. And so in order to like build all this, the best thing to know is that it's always going to be polymerizing the 5' to 3', or like adding onto the 3' end. Yeah, and so a lot of the diagrams are like this because this is just like the dashed lines, and this is what I was just talking about earlier, where it can add continuously to the 3' end, but over here, since it's opening up, like it's not able to just, not all the 3' end is exposed over here. Whereas on this side, the 3' end is always exposed as it's like continuously being exposed as it's opening up. And this is just like a more detailed view with other pieces involved. And so the lagging strand is going to have a little bit more involved with making it. And so, as I said before, there's like an RNA primer, but since it's opening up in this direction, it needs to add multiple like continuous RNA primers as it goes. And then it'll fill in, the DNA polymerase will add DNA on until it goes to the next RNA strand. And these fragments of DNA are called Okazaki fragments, named after the guy who discovered these. And then this is a sliding clamp, which just holds the DNA polymerase on. And then yeah, whenever the DNA polymerase is used, sliding clamp is needed. And then RNase removes the primers, and DNA polymerase can go back and add on whatever is missing. And then at the end, there's going to be between these, there's going to be like DNA pieces at the very end of this, but they're not going to have like there's going to be like a phosphodiasporan that's like missing. So they have to have like a DNA ligase to seal the mix in between. And so the thought process behind like figuring out which one's leading or lagging is that the DNA strands that are new are always going to be synthesized 5' to 3', always adding to the 3' end. And the direction of the fork is starting from the origin, moving outwards in both directions. Yeah, so you just look at both directions of the parents and then draw in what the newly synthesized strands would be, and then you can figure out. Yeah, so you can see right here, this is a really good diagram actually. And so you can see from here that this is like the middle of like a origin replication right here. There's like a bubble, and this is like one side, and like on the other side there's a mirror image, but this is kind of zoomed in. And so as you can see, if you just label your sides like 3', 5' on the parent ones, you can figure out, okay, this is a 3' end, this is a 5' end. As it opens up in this direction, is the 3' end in the direction of the replication bubble, or as it opens, is there like a piece that's like exposed here afterwards that needs to be added onto? And that would be the lagging strand. And then this is the leading strand. And then, yeah, this is literally the same thing. And then origin replications, this is just a more detailed diagram showing basically the ORC and like connecting what you learned about earlier. The ORCs are going to fire off in both directions, and helicase is going to, like the MCM complex you learned about, is going to go off in both directions until they meet another bubble. And this is just like a more detailed look as well on one side. So you have like different proteins, like sliding clamp, like we talked about, has to be holding on to DNA polymerase in both directions. But this is the leading strand, so it's just continuously being synthesized. And then the RNA primer right here, right here, they're being in a lagging strand. There's like multiple of them rather than just one, because of it being opened in the opposite direction of the synthesis of DNA. And clamp loader is just another protein involved with the sliding clamp, which puts the sliding clamp onto the DNA. And DNA primase makes the RNA primers. Yep, red is parental strand. And then this is the same thing, just talking about sliding clamp, helicase, sliding clamp loaders on this picture. Also, when single-stranded DNA is like exposed, but it's not being like replicated just yet, there's a single-stranded DNA binding protein, which will protect any exposed single-stranded DNA, as shown here. Oops. And yep, this is, yeah, so this is just a zoom in view of all the machinery. And then here's just a summary of what I was just saying. Yeah, it's single-stranded DNA is fragile. Any questions about this stuff? Mhm. Here?
Visualizing how it works into fragments. I think the professor mentioned that it's tethered to it, that's why it stops, or like, but I'm having trouble with visualizing that. Is there a way?
Yeah,
So as you go like this way, basically, so like there's synthesis or the bubbles being opened this way, right? And this way. So this is the lagging strand, right? So what you want to do is, if you look at 5' and 3' of the parental strand, right? If you were to like label these, this would be 5' and this would be 3', right? See if there's, oh, it just really right here. Yeah. So from that, you can figure out, okay, like the numbers on here. But once you get the numbers, you have to think about, okay, this direction is where it's opening up, right? And I think it's easier also to find like the leading strand first. So this would be the 3' end, this is the 5' end, right? And if it's opening up this way, the number one thing to keep in mind is that you're always adding to the 3' end of a DNA strand. So this is the 3' end right here of the growing one over here. That you can figure out from the 5', right? So 3' since they're complementary and anti-parallel. So since this is 3' and it always adds on to the 3' end, as this opens up continuously, it's gonna be able to just keep adding on. Whereas on this side down here with the leading or lagging strand, as this opens up, the exposed end is not the 3' end, it's the 5' end, right? So what's gonna happen is as you open up, you'll have like these strands, right? So let's take this for example. You have these strands right here. As you open up, there's a 5' end right here, and there's gonna be more and more exposed DNA. And as there's more and more exposed DNA, right, you can't just keep adding these DNA nucleotides on. And so you're gonna have a big problem with being able to synthesize the rest of this DNA strand. And so in order to overcome that, what happens is that the RNA primase will just, or DNA primase will put down like an RNA primer right here. As it opens up, it'll put a little one down, and then, or like right here actually, and then the DNA polymerase will start to add on. So like this will be like where the new Okazaki fragment would be, right, or the RNA for it, and then the DNA would start to add on over here. So like this is the beginning of one right here, like this would be like RNA, and it has to add onto the 3' end, so that's why they're growing like this. Does that make sense?
I can also try drawing it for you after.
Mostly, like, is there a video to watch? Because I put.
Yeah, I know, I tried looking for one, I think is there one in the movies folder? Did you check
I'm not sure.
So I'm probably gonna end up going through it.
Okay, yeah, there might be one there, but if not, just like look up like flagging student
Yeah.
on YouTube, but yeah, I can try drawing.
Maybe I should make a video.
A little bit, like, don't like the RNA primers like fill in the gaps in the lagging strand leaves behind, right?
Oh, the DNA ligase.
Ligase, yeah.
Yeah, ligase. Yeah, so, oh no, sorry. So once the so the primers need to be removed, right? Because they're RNA, and so DNA polymerase. After the primers are removed, it uses 3' end of like another Okazaki fragment to fill in that where the nucleotides of the primer left behind, right? And so then there's that one little nick at the end that DNA ligase.
Yeah. Yeah, maybe, yeah. I think I should probably make something.
What?
I should probably make like a video
Of
or
what?
something of like me
Yeah.
drawing it out and stuff, yeah. But yeah, we can answer more questions at the end too. So moving on, we'll go, unless you guys have more questions right now. Okay. So based on this knowledge right here of adding onto the 3' end, the issue with our chromosomes is that at the end, since they're not circular, at the end of this double-stranded DNA, the bubble is going to get to the end and there's going to be this RNA primer that's going to be put on by the DNA primase. And this would be like the beginning of like an Okazaki fragment, right? But the problem, and same thing over here. But the problem is that this system relies on another fragment being built from behind because there's like more of this lagging strand template. Usually there is, at least for these other Okazaki fragments that were built up here, there was more DNA down here, but the problem is when you get to the end, there's no more DNA. So there wasn't like an RNA primase down, or DNA primase down here, putting down RNA primer for DNA polymerase to build off of and then replace this RNA primer. So what ends up happening is that there's this gap that gets there ends up just being a gap and the RNA is removed. But because of that, some of the DNA, the next time it replicates though, so yeah, this excess DNA is going to get cut. So the next time it replicates, it's going to lose a little bit of information. But even if it's like, you know, like 8 to 10 nucleotides, it might not seem like a lot since that's like the length of a primer. But this, you have to keep in mind that this is like every S phase, which would be every time a cell divides. And there's like, the cells divide like millions of times in your body. So over the course of time, there'd be a lot of information lost. So in order to get around this problem, there's these things called telomeres at the end of the chromosomes, and they're specialized DNA protein complexes. And they are basically non-coding DNA sequences that repeat over and over again. But when they become too short, the cells will end up dying to prevent there being like a loss of information. So in order to do this, telomerase is going to solve the end replication problem by adding on with a RNA template of its own. It's going to help add on some DNA nucleotides to the end of the like of it. What is it?
The one with the lead.
Yeah, the lagging strand, right? Lagging strand.
Well,
fragmentation.
it makes that top strand longer, right? So that it can add another.
Oh, yeah. Right, yeah. So it ends up like, because it has this, so it brings over this template. And so because there's a 3' end available right here, when there's a special protein that does DNA polymerase based on RNA information, and it's going to do reverse transcriptase. And it's going to do RNA templated DNA synthesis, and it's going to add on these like random repeated nucleotides onto the 3' end of the lagging strand, parental strand. And then because of this, even if there's like a loss of information, right, when it goes to like, when there's when the DNA primase goes to put down RNA primers on the DNA, it's going to be able to put them down far beyond the point where there's like actual information in the DNA itself. And so because of this, there'll be a lot of extra DNA at the end of the chromosomes called telomeres. And these telomeres are basically just these non-coding DNA, repeated DNA sequences. Does that make sense to everyone? Mhm. Yeah.
Like, how does that like add? How does that make that longer? Because I understand the fact that's bringing like another like strand to the leader strand to stop it from like being reduced, but like How's that vaccine the pregnancy?
So this is the parental strand right here. Right? And so because the whole problem is that we can't, this is the newly synthesized strand. The whole problem is we can't just keep adding directly onto this because it's the 5' end. But with this parental one, it's the 3' end that's exposed. And since we want to add on to the 3' end of nucleotides, that one is gonna allow us to put nucleotides onto that end directly. Does that make sense? Or because the thing is, honestly, like all this material just comes down to you only add onto the 3' end. So keeping that at like the core of like it sounds easier than it is, but like that's like what the problem sets for it. Like obviously all this, but at the end of the day, like I said, like it's just when you keep it in mind that you can only add to the 3' end, and look at it that way. Knowing that this is the parental strand right here, that's why the 3' end right here is where you're going to be adding onto that. The DNA polymerase can come in and add on nucleotides to the 3' end. And so because of that, that's why. Yeah.
Thank you.
Does that make sense?
Yeah, actually, yes.
Any other questions? This is a really hard concept. This is honestly probably the hardest concept of this week. That's it.
You
The,
can stop it right now to do a question. And we can also answer